Okay, I'll assume that everyone can hear me. So I'll go ahead and get started here. Our topic for today is meta-analysis. So my do this here. My outline for today is I want to talk about the methods of meta-analysis and how we go about combining studies and then I want to actually use 3 different examples of meta-analyses to illustrate some of the issues and as well as the methods. And then I'll end up with a brief summary. So meta-analysis combines the published results of several studies, and primarily it's used to overcome the sample size issues that you might have with small studies and whether they are valid or not, and then to estimate the so-called true effect size of some treatment or therapy or whatever. So meta-analysis that I'm going to talk about today is based on summary data, that is data that you can get from published studies. There's also what's called patient-level meta-analysis, but that's combining actual data, individual patient data from several studies. And you don't see too many of those because you have to have access to the data. And if you want to, do that, probably not too many people are just going to send you the data. So it's usually done by people who have been involved in the studies one way or another. So meta-analysis has appeared in published studies in an increasing number over the past few years. However, many of the top journals, for example, like the New England Journal, actually don't publish them. And there's a reason for that, which we'll get into. So let me go through the methods of doing a meta-analysis. And again, I'm talking about doing a meta-analysis from published studies. In other words, you're basically getting summary data. So it begins with a systematic review of the articles that deal with the topic of interest. And so to do this, you need a librarian, and then you have to come up with keywords for your literature search. And so there can be, a fairly large number of articles, and I'm going to illustrate that later on with a particular study. But once you have your keywords together and the literature search is done, then you need to go through those articles and find out which ones are actually applicable to the question that you want to ask. And so you need at least 2 independent abstractors to go through the articles and come up with a list of ones that apply. A very important concept in meta-analysis is the idea of fixed versus random effects. And what we mean by fixed effects is that the effects on of a treatment on some outcome or whatever are attributed to a finite set of levels of a factor or variable that are of specific interest. So like you might have 2 different treatments and other variables like that you might be interested in. Random effects, on the other hand, are effects attributed to an infinite or at least a very large set of levels of a factor of which only a random sample appear in the data. And so in meta-analysis, typically studies are going to be treated as a random effect. And so we'll illustrate that a little bit. So random effects from a given cluster, so it may be correlated. In other words, they're not independent. And so the correlation may affect the model parameters and standard errors if not accounted for. And generally, including random effects will result in larger standard errors and thus larger confidence intervals. So in terms of meta-analysis, the published studies or the studies that you come up with in your literature search are considered a random effect because there are only one set of many comparable studies that may or may not have been published. So typically studies in a meta-analysis are treated as random effects. So let me illustrate that again with an example from the Courage trial that was from several years ago, 15, 20 years ago. And so in the Courage study, we're looking at a composite outcome of death or non-fatal MI or non-fatal stroke, I believe. But at any rate, there were 50 hospitals involved, but these hospitals came from different health care systems. So there were 16 Canadian hospitals, 19 US non-VA hospitals, and then 15 VA hospitals. And so there were some 2,200 or almost 2,300 patients enrolled in this study. And this is a little schematic to illustrate the concept of fixed and random effects. So we have the different healthcare systems, and then within each healthcare system, you had any number of institutions or sites, participating sites. And then within each site, you had a different number of patients, and then patients were randomized to either receive percutaneous coronary intervention or optimal medical therapy. And so the fixed effect in this case is the treatment of PCI or OMT. The random effects, of course, patients are always random effects. The sites are random effects because these would only be a sample or a certain selection of sites within each healthcare system. So in this case, we have really three random effects: patients within sites within healthcare system. And so that's the concept of what we mean by random effects. And so in meta-analysis, then the studies are going to be treated as a random effect. So the validity issues that we have in meta-analysis are, as I mentioned, random or fixed effects, but primarily the main one is, the really important one is what is referred to as heterogeneity. In other words, that's the idea of variability in results. In other words, if you have, say, a dozen studies in your meta-analysis, they're not all going to have the same treatment effect, say, for example, on the outcome. And so the variability, how do we account for variability within each study? And then other issues, there might be one study that's really influential that, and how do we determine whether that's true or not? And then also the order of the studies or the year in which you are published. In other words, is there accumulation of evidence, or do the studies, the year in which the studies were done or then published, how long a time between those studies? And then what we'll look at is also study bias. So when we talk about homogeneity or heterogeneity in meta-analysis, as I mentioned, we're going to find differences in the results of individual studies. And the basic assumption that we would like to make is that any differences are due to just chance or random sampling variability. And the issue is that that's not always the case. And so we're going to look at some studies where, and statistics to determine whether the differences are due just to chance, random sampling variability, or whether the differences are due to something else. So in terms of bias, probably the main one is selection bias. In other words, you get a large list of publications that apply to your study question, but you may not get all the publications, or you may not get all the studies that were done, because some studies don't get published, particularly negative studies that don't show any difference between, say, treatments or therapies. Or maybe there's a location or language issue. Most meta-analyses get publications that are published in English. And then we've talked about heterogeneity a bit. And then the data within each study. How good is the data in each study? Maybe you have some incorrect data, or they've done the wrong analysis, or something like that. And then just artifacts like measurement error. If you're have an outcome that is defined by, say, patient-reported outcomes, then there's a lot of issues related to the reliability of those outcomes. As far as the particular statistical methods are concerned, it really depends on whether you have a dichotomous outcome, which a lot are, either like dead or alive, yes or no, positive, negative. So in that case, our analysis is going to be a logistic regression, creating an odds ratio or risk ratio, or sometimes it's done on the log of the odds ratio. If we have a continuous outcome, then we're looking at differences in means or rates. And the primary method, which I'm not going to go into the details of that, is a method for analyzing studies as random effects. In other words, we're pooling the statistics of each study in our meta-analysis, and we want to come up with an overall or pooled statistic that tells us what the true effect is. And the particular method, Dersimian and Laird method, is one that's virtually used 99.9% of the time to do this. And then a statistic that we're going to focus on, because this is, at least in my opinion, is probably the key statistic in a meta-analysis, is the I-squared statistics. And that's going to tell us how much of study heterogeneity or how much variation in the odds ratios or mean difference is attributable to differences between the studies. In other words, is it basically going to be due to random sampling variability, or is it going to be to actual differences in the studies? And the I-squared is presented as a percentage. It can range from 0 to 100%. The larger the I-squared statistic, the more heterogeneity. And what that tells us is whether the combination of studies that we have in our meta-analysis is valid to put those studies together in a meta-analysis. Then what you'll commonly see is what's called funnel plots. This is a plot that gives us an indication of potential bias in the studies, what's commonly referred to as publication bias. You may or may not see the jackknife or the leave out one analysis, where you do the analysis however many times you have, or how many studies you have in your analysis, and then you do that a number of times, leaving out one of the studies and seeing if one study is making a difference or is influencing the overall results. And then another method that we often see is what we call meta-regression. In other words, we introduce into the analysis covariates that might affect the outcome. So for example, if your studies comprise a number of years, we could look at study year to see if that affected the outcome or other covariates like that. Okay, so what I'd like to do then is to sort of illustrate these concepts with particular meta-analyses, and I'm going to do 3 of them. And the first one is a meta-analysis was published some years ago, looking at the effect of PCI, percutaneous coronary intervention, on long-term mortality. And this arose because, and maybe it still is an issue, but some years ago there was controversy regarding the impact of PCI versus medical therapy on long-term survival in coronary artery disease patients. And the trial that I mentioned before, the COURAGE trial, was a large study that found no evidence that PCI as an initial management strategy reduced the risk of death or MI, non-fatal MI, or death alone when added to optimal medical therapy. So there was a study, a meta-analysis of 16 published randomized trials and 1 abstract that looked at PCI versus medical therapy. And that study concluded that there was a benefit of PCI for all-cause mortality. However, it turns out that the selected studies mixed the patient types. In other words, there were not just CAD patients, but there were also patients categorized as post-MI or acute coronary syndrome patients. And then also, the meta-analysis omitted a major post-MI trial, the occluded artery trial. And so what we did is we took the data, because you actually have the data in the meta-analysis paper, and we reanalyzed it, distinguishing trials of different patient types. And then we did another analysis where we included the occluded artery trial. And so this was the initial presentation by the meta-analysis of stable CAD patients, but it also included 5 trials that were patients of different type, post-MI patients. And so they found that there was a overall, by combining the results of these studies, an overall benefit for PCI. In other words, if you were, if the odds ratio here was less than 1, then it favored PCI. If it was greater than 1, then it favored medical therapy. And so you see that they did both a random effects and a fixed effects model, and you get the same point estimate, but you can see that for the random effects model, the confidence interval is wider than the fixed effects model. And their I-squared for heterogeneity was 17%, which indicates that there's some difference in the studies. One thing I will say, and I'll probably say it more than once, is when you look at a meta-analysis and you look at the I-squared statistics, forget about the p-value for it, whether it's statistically significant or not. Look at the actual value of it, because the p-value, whether it's statistically significant or not, is dependent on the number of studies, not on the number of patients. So this was the results, and they concluded that there was benefit for PCI over medical therapy. And one thing I want to point out in doing a meta-analysis, you can see that there's one study here that for either group, there's no events. And so the typical meta-analysis software that you can use, of which there is a whole lot and a lot of free downloads of meta-analysis software, this particular study would be excluded because there are no events. But you can still see there's 100 and some patients that have no events. So that's kind of one drawback where you have studies like this where you don't have any events. So they did their meta-analysis and arrived at the conclusion that PCI had a positive effect. That is, that it reduced long-term mortality more so than medical therapy. So what we did is we took their data and looked at differentiated studies that distinguish by patient type, either chronic coronary artery disease patients versus post-MI patients. And so we did meta-regression of studies grouped by patient type, and then we calculated the I-square statistic to assess trial heterogeneity. So this is our results, that there was a significant, in the meta-regression, there was a significant treatment by patient type interaction. And our meta-regression estimated the mortality odds ratios for the CAD patients is 0.9, which the confidence interval included 1. So that would argue for no benefit of PCI for those type of patients. However, for the post-MI patients, the odds ratio was 0.5, which the confidence interval did not include 1. So that would argue for a benefit of PCI for those patients. And so we redid the forest plot and divided the studies into the CAD patients and the post-MI patients. And you can see that there's some, you can see the difference here, that the I-squared for the CAD studies is 0. In other words, that means that the variation between the estimates here, the odds ratios, is just due to random sampling variability. On the other hand, for the post-MI patients, the I-squared is now over 20%, which would indicate that there's something other than just random sampling variability or chance that's that's the reason for the difference in these estimates. And so then what we did, as I mentioned before, we added the OAT trial, the occluded artery trial, and this looked at the same treatment, PCI versus medical therapy, but it was only for post-MI patients. And so what they found was that the study, again, a large study, did not support the use of PCI for these type of patients. And so what we did then is we took the data from the OAT trial and we added it to the post-MI and recalculated or redid the meta-analysis. And so you can see here that now when we add the occluded artery trial, that it really kind of negates the PCI effect, in that the confidence interval now includes 1, so that would argue for a non-significant effect. And the I-squared now, by adding this trial, goes up to over 47%. So now what that's saying is that the combination of these studies may not be legitimate, or pooling the results from these studies may not be giving you a valid result. So the meta-analysis, redoing the meta-analysis, we found no evidence of a PCI benefit for the CAD patients. Without the OAT trial, there is an evidence of a PCI benefit for the post-MI patients, but if you include the OAT trial, then it minimized the benefit of PCI on mortality. So the point here is that the pooling of different patient types could lead to the wrong conclusions or invalid results. And these are, okay, so this is just sort of summarizing what I've kind of already mentioned. Okay, so then the second one that I want to focus on is a study that looked at mortality in teaching versus non-teaching hospitals. And so I wanted to sort of show you the literature search statistics here, that we use 32 search terms, which is a lot. And so there turned out to be over 32,000 potentially relevant citations identified. So this is a huge number. So we had two reviewers that went through the 32,000, and they came up with 822 articles. And then they went through it again, and with this time with 3 reviewers, and another 582 were excluded. And then because that's a lot of studies, they went through it again, and they found that actually there were only about 250 that had some teaching versus non-teaching comparison. So you can see that you may start out with a lot of studies, but not all of them are particularly relevant to the question you're asking. And then it turned out that out of these 249, that not all of them actually had data that you could use in a meta-analysis. So we came up with actually just 100 studies where there was teaching versus non-teaching data that we could actually use in a meta-analysis. However, the study outcome, mortality, was defined differently in studies. So 61 of them was focused on in-hospital mortality, 35 on 30-day mortality, that is 30 days post-discharge from the hospital, and then 4 of them looked at greater than 30 days. So here, the issue is we're going to do the analysis on mortality, but we have different definitions or different time spans for mortality. And the studies themselves span 21 years, which is quite a length of time, and involved a lot of patients, 26 million patients. So the results turned out to show, using the random effects odds ratio of 0.9, and the confidence interval did not include 1, so we would conclude that the teaching, for a teaching hospital, the mortality rate was significantly less than the non-teaching rate. However, as it also turned out that virtually all of the variability in odds ratios between the studies was due to study heterogeneity. In other words, it was virtually 99%. So that really, in a sense, sheds doubt on the conclusion that there was a teaching, or there was a benefit of teaching hospital with regard to mortality. And their use of the funnel plot shows shows this. So this is what a funnel plot is. It plots the log of the odds ratio, or it would be, if you're doing a continuous variable, the difference in means or rates, versus the standard error of the log of the odds ratio. And so what the way that to interpret a funnel plot is that you plot each of the ratios here for the log odds and the standard error for each study. And if it falls within the 95% confidence limits, then we can conclude that there's no bias in the study. But in this case, you can see that a large number of studies with small standard errors, that is, these would be the larger studies up here, there's a lot of variability here. In other words, they're not falling within our confidence intervals here. So ideally what you would want is the larger studies to fall up here and the smaller studies to be down here with the larger standard errors. But it turns out that for this study, that's not the case. So the problem in this study is, first of all, that combining them is questionable because of that nearly all of the variability in the odds ratios between teaching and non-teaching was due to differences between studies. So even though our endpoint was mortality, the time of death was not taken into account. In other words, we had 30-day or in hospital 30-day mortality and greater than 30-day mortality. So it looks like it would have been important to distinguish between those time points. Okay, so the third and final study that I want to focus on is actually a fairly good study, and this is not comparing. This study doesn't compare particular treatments, but it's looking at a particular patient type, patients with angina but without obstructive coronary artery disease, and it's gathering studies to look at what the rates are. In this case, the primary outcome was the composite pooled incidence of all-cause death and non-fatal MI. So I wanted to just present some of their methods and some of their results here in terms of their what they said that they were doing. So what they did for each study, they calculated the incidence and its 95% confidence interval for each of the predefined outcomes. That would be the composite outcome, and then each one of the components separately. And so what they did is they expressed the incident as ratios between the number of events and the number of person time, in this case, years at risk. And then they indicate here how the confidence intervals are calculated for the incident. And then so they use the random effects models to estimate a summary of the incidence. And they also then, as I've talked about before, they performed a jackknife as a sensitivity analysis for each outcome to evaluate the robustness of the results. So as I mentioned, a jackknife is done, leave out one, and then recompute the outcome, in this case, the incident per person years, and to see if there's any one study that is affecting those results. So here is their forest plot. They had 54 studies, And so the incident rate per 100 person years turns out to be right at 1. But you can see here that there's a large degree of heterogeneity. The I-squared is over 90%. It was reported as 91% in the study, but when I actually recalculated the data, it was a little more than that. But regardless, there's high heterogeneity in their study here. So what they concluded was that the incidence was about 0.9 or almost 1 per 100 person years. And then they did state that there was considerable heterogeneity among studies. But they also then did a meta-regression to their credit. And so they found that the primary outcome was associated with dyslipidemia, diabetes, and hypertension. And of course, they would have looked at each of those separately, and they didn't look at them in combination. So in doing the meta-regression, here, this is the kind of plots that you'll often see to show the association between the prevalence of the covariate and the, in this case, the incident of the composite outcome, all-cause death, and MI. And so what these plots represent, of course, the straight line is the regression line, and then the circles are sized according to standard errors. So the smaller the circle, the better the standard error. The smaller the standard error, the larger the circle, the larger the standard error. So a large standard error would probably indicate a small study that didn't have that many patients in it. So you can see that they find a significant regression line. In other words, the regression line is not statistically zero for dyslipidemia, hypertension, and diabetes. So because you have their data, so I took their data and sort of reanalyzed it and looked for studies that were homogeneous, that is, where the I-squared was small. And I took 10% as sort of a cutoff here. I think 25% is sometimes used as a cutoff for a small degree of heterogeneity. So I then ordered the studies from lowest to highest effect size. And so the forest plot would look something like this. And so by looking at how these studies sort of grouped together, I came up with at least 4 or at least 3, maybe 4 different groups of studies where the difference between their estimate of the incident was due just to random sampling variability. And so what you can see here is that we have a group of studies where the incidence is quite low, about 0.4. And then other groups of studies where it's higher, 1.1 here and 2.1 for these sets of studies, although the constant intervals do overlap here. So, and then the last 3 studies where the incident is quite large, of 5. So the question is, we have, or the issue is, we have at least 3 different estimates of the incident rate here, from really small to quite large. And so to say that the overall incident, to conclude that the overall incident rate is 1, which was the pooled estimate from all of these studies taken together, it probably isn't a correct statement to make. The authors, though, of this study did do, I think, a fairly decent job, though, because they said that there are 2 things here. One, as far as the outcome, they did say this appears to be extremely variable. So they did recognize the large degree of heterogeneity between studies. One possible explanation for that is they also found that angina without obstructive CAD is inconsistently defined. So that means that among all the studies that they all didn't define their patient group in the same way. So that also contributes to heterogeneity and calling into question as to whether or not you can combine this particular set of studies. So all in all, this was a pretty good study, although there were some things that are left out, and in particular, what is left out of a lot of meta-analyses is dealing with a large degree of heterogeneity. So it's really important to look at the studies that you put together, and when you're reading meta-analyses, the first thing you probably want to look at is the I-squared statistic to see what it is, how high it is. Okay, so as far as this study goes, they did mention that there was high between study variability. They did find through meta-regression some clinical comorbidities that were associated with the outcome. But on further investigation, I found that there were at least 3 groups of studies with non-overlapping confidence intervals. And so is there an explanation for this? Is there demographic, other clinical variables that identifies groups? Or as they mentioned, there was there was differences in the definition of their study population. So that probably also contributed to the high study variability. Okay, so in summary then, meta-analysis is a tool for combining results of several or many studies, but it requires methodological rigor and careful inclusion of homogeneous trials and avoidance of selective exclusion. So in the first meta-analysis that we looked at, the one looking at PCI versus medical therapy, there was some evidence of excluding a trial that should have been included. Whether that was intentional or not, you don't know. But that's one of the reasons why meta-analysis are somewhat suspect, because you can select the studies that sort of agree with your preconceived notions. Then also we need a common definition of the outcomes for it to be valid. And for most meta-analyses, you want to treat study as a random effect. Now, if the I-squared is equals 0, then random and fixed effects are essentially equal. They would have the same confidence intervals, because the randomness would not be taken into account. Okay, so as far as evaluating meta-analyses, what you want to look at is patient populations. Are they mixing patient populations that are really different? Do they do random or fixed effects? Although that's pretty much anymore. You see one, or you see random effects, or both of them are done. And then heterogeneity, as I mentioned, probably the first statistic that you would want to look at is the I-squared statistic. And this should be presented in either the narrative, or it's usually also presented in the forest plots. And then, are there factors influencing the results? you know, the time interval of studies, where they were published, whether it was a clinical, a randomized trial, or was the study observational. Sometimes you see those two mixed, sometimes they're separated, but there's usually a big difference in randomized trial results and observational studies. And then as far as doing meta-regression is something you probably want to do or want to at least look for, but the drawback there is that not all studies include the same covariates to look at. In other words, you might find, say, for example, diabetes as a comorbidity that you want to include in a meta-regression, but you know, some of the studies may not identify patients with diabetes, so that poses a problem in that you may not be able to include all the studies in your meta-regression. And then finally, I guess my main point I want to make here is when you look at a meta-analysis study, be very skeptical. So I have some references here. The first two are a couple of the studies that the published studies, meta-analysis that we looked at. And then the last two are more methodological studies. I would recommend the first Higgins one there, the one from Statistics in Medicine. It's actually fairly readable. It does show how heterogeneity is calculated, but it's really a good article on looking at heterogeneity in meta-analyses. So I want to thank you again for your time, and if there's any questions that you have or anything that you would like to address. Let's see here, I have one. Oh, okay, so this is a question about combining observational or cohort studies and randomized studies. So I believe that most of the studies in the teaching versus non-teaching one were observational. There were some that were, I believe there were some that were randomized trials, but not very many. So in that particular case, most of them were observational studies. So I forgot to mention that's one other thing that you could look at is the type of study that it is, because obviously randomized trials are a stronger study than observational studies. So let's see. Okay, another one here. Okay, yes. So there are, the question is, are there any study quality assessment tools specifically for meta-analysis? Yes, there are. I didn't list any of those, but I can. I'll put out some references, and you can probably just go online or Google meta-analysis and find some of those. But some studies and more recently have made attempts to look at the quality of a study and to rate the studies, to give a study, all studies in the meta-analysis, a quality assessment. And some of these are based on the sample size, based on whether it's a randomized trial or a observational study. The ones that you mentioned there, the MSTAR and ROBUS for systematic reviews, those could be used for a meta-analysis as well. So typically what's done is they give you a table of the quality for each study in the meta-analysis. But there are methods for actually incorporating the quality assessment into the meta-analysis. As I say, I'll look up, get together some references and put those out that you can look at. Okay, so let's see. Okay, so yes, this session is recorded. They put it out on YouTube, and it'll be available, I think, in a few days. Let's see. So another question here. Please discuss how to distinguish secular versus cohort trends in meta-analyses. I'm not sure I've seen studies that actually try to distinguish that. I think maybe of one. So basically, you would look at most are just probably cohort trends, but you could look at the study date when the study was published or when the study was done, or look at them over time as they were published from the least recent to the most recent. And so another question was, when I did the grouping in the last study, did I try to assess? I actually didn't, but that's something that could be done. That I could look at each study, go through each study, and see if there was something common to those studies that would make them more similar. If you're actually doing the meta-analysis, and do that sort of thing, you definitely want to do that. But I was just looking to see if I could come up with different groupings, different estimates of the outcome. There was one that I did some a couple of years ago where it turned out that they actually did identify differences in patient types. Okay, let's see. Let's see what else here. Oh, okay, so we'd like to see the papers again. Let me see if I can do that. Oops, let's see. Oops. Let me get back here. Okay, so here are the references. Actually, I'll send the references out also to the CTSA and send this out to everyone as well. And then my email address, paul.kolm@medstar.net. Okay, well, thank you for your time. And we'll, in February, the session will be on Bayesian analysis, so it'll be a different perspective than we've looked at before. So thank you for your time, and we'll see you next month.